Okay, so welcome again. My name is Reena. So this is our second lecture regarding the comparative anatomy of integumentary system. So during development, you are having this ectoderm. Below this, you have neural tube, and below this, you are having this notochord, and you are having endoderm here, and in between this ecto and endoderm, you are having mesoderm. Okay, so this is in your embryonic stage. Mesoderm is divided into different zones. Okay, let's just consider this is your notochord, okay, which is formed from the primitive stake. So this is your notochord, and then surrounding to this notochord is present your mesoderm zone. This mesoderm, which is lying just beside the notochord, it is called paraxial mesoderm. The para means side, axial means axis. So the notochord which is lying just beside the axis, this notochordal axis is called paraxial mesoderm. And then you are having another mesodermal zone, okay, both side. This is your intermediate mesoderm. Now the rest of the mesoderm, which is lying beside this intermediate mesoderm, is lateral mesoderm. So the somites that we are going to study about, it is going to be formed from this paraxial mesoderm. What happens is this paraxial mesoderm, okay, so this is your notochord, okay, so this paraxial mesoderm is going to get segmented into somitomers, okay. This is the caudal region, this is the caudal region. So here you are having some caudal plate mesoderm. They don't undergo segmentation, and rest of the mesoderm are lying below this caudal plate mesoderm are going to get segmented. These somitomers could be around 44 in number. So yeah, if we see them in a cross section, they are having a triangular cross section like this, okay, and they are having a cavity inside also. This somite is going to be actually having three zones. So this first, this first zone, the uppermost zone, this is going to be called dermatome. The second zone, it is going to be called myotome. Third zone is going to be called sclerotome. Okay, this dermatome is going to make the dermis part of skin and also the hypodermis, which is actually your subcutaneous tissue. And the myotome part of the somite, it is going to make all the musculature of your skin, okay, the musculature like tongue also. Assume that this part is your paraxial mesoderm, okay. The upper two regions were your dermatome and myotome, and this third part is your sclerotome. This is going to move around the neural tube, okay, and around this neural tube it is going to make all the skeletal elements like vertebral column, and then you're having ribs. It is going to form these elements, these skeletal elements. Okay, so this was your paraxial mesoderm, and then talking of intermediary mesoderm. So actually, this is nephrogenic tube. What does it make is it makes kidney and gonads. This green area is here, okay? So what it is going to split into two region. First one close to the ectoderm, second one close to the endoderm. Okay, the one which is closer to the ectoderm, it is going to make the somatic mesoderm, and the one closer to the endoderm, it is going to form the splanchnic mesoderm. Okay, so scales. Actually, we are studying about the comparative anatomy of integumentary system in vertebrates, right? So scales are of two types. Depending upon the origin, like from where they are originated, they are of two types. First one is epidermal scale, and the second one is dermal. In this case, we are having stratum germinativum, or you can also call it stratum germinativum, or you can also call it stratum bacilli. This is going to get cornified into scales. The epidermal scale, as you already know that the periodic shedding of the scale happens in the snakes. Okay, so snakes, they come under uh, reptilia, prominent epidermal scale, and then some epidermal scale, they are permanent, they don't shed off, and those scales are called cutes, and they are found in turtle, crocodiles, etc. Okay, so this was your epidermal scale. And talking of dermal scale, obviously they are going to be formed from dermis. They are mesodermal in origin. They are actually best defined in fishes. And the exception in this case, like some fishes are there which are not having any dermal scales. Those are electric rays and catfish. We will study about these epidermal scale when we come to the reptilia part. On the basis of vertebral hierarchy, for example, in vertebrae, you are having first class that you are having is cyclostome, and then you are having placodermy. And we are not going to study about placodermy because they are extinct species. So we are directly diving into chondrichthyes, and then osteichthyes, and then amphibia, then reptilia, then aves, and finally the mammals. Okay, cyclostome, they are not having any scales. We are studying directly from the chondrichthyes. As I've already told you that we are not studying about the placodermy here because they are extinct species. So this type of scale they are having is cosmoid scale. You just have to memorize this name of the scale that primitive fishes used to have. In this lecture, we are going to see the comparative anatomy of scale in living fishes. In living fishes, the scales are of four types. They could be of placoid scale, they could be ganoid, they could be ctenoid, they could be cycloid. Chondrichthyes, they are having placoid scale. So we will see how placoid scale are developed in these chondrichthyes. Let's just see how the scales are going to get developed from the dermis. Let's just assume this as an epidermis or stratum bacilli. So what happens is you're having some mesodermal cell here. They are going to get aggregate just below the stratum bacilli or stratum germinativum, and it is going to push the epidermis upward. Okay, so the first step is mesodermal cells are going to aggregate just below the stratum bacilli, and they are going to push the epidermis upward. Your epidermis has gone something like this. Okay, so this has become your dermal papillae. So some cell out of these mesodermal cell aggregates, they are going to change into odontoblast. They're going to differentiate into odontoblast. And the function of this odontoblast is they are going to secrete dentine just below the epidermis. Okay, some cell above the epidermis, just above the stratum bacilli, they are going to get differentiated into ameloblast, and they are going to form an enamel-like organ, which is actually going to secrete vitrodentine over this dentine, which has been secreted in the dermal region. So basically, this is your vitrodentine. Below this, you are having secretion of odontoblast, that is dentine. This is your dermal papilla. Cells of stratum bacilli yet there, because the dentine has not protruded outside the epidermis. A pulp cavity is going to form for the growth of the scale. So these are some blood capillaries. The star-shaped thing that I'm making is odontoblast. They are actually going to secrete the dentine. So for the nutrition of this odontoblast, we are having this pulp cavity, and this pulp. Okay, this is your pulp cavity. This is your vitrodentine, which has been secreted from the ameloblast, and then this is your dentine, which is secreted from the odontoblast. So basically, this is your placoid scale, and the base of the scale is called basal plate, and it is formed from the mesodermal cell, which are actually aggregated here, and they get transformed into odontoblast as well. So in the dermal region, I have already talked about the connective tissue. Okay, I have already told you that there is a high amount of reticular connective tissue that is present in the dermal region, and from reticular tissue I mean that they are having a highly fibrous connective tissue there. So I have already told you about the collagen and elastin, but another type of fiber that is going to support the basal plate is sharp-lace fiber. So these lines that I am making here, rod-like structure, they are actually sharp-lace fiber. So after the scale is fully grown, it is going to burst the epidermis and going to protrude out from this epidermis. So at last, the fully grown scale that you are going to see in the surface of chondrichthyes epidermis is like this. Okay, so this is your scale, and here is your epidermis. So it is something like this.